a question. And then voting, assuming we have a quorum today, we will vote in the negative, meaning if you do not wish to provide your approval, then vote. Or if you wish to abstain, then vote. Otherwise, the assumption is that you agree with any motion brought forward. Okay. So want a quick review of the agenda before we get going. So we'll have our call to order and introductions, our quorum certification. We're going to review and approve the changes to the big roster, review and approve the minutes from the last big meeting, have some time for public comment, and then several items to take care of, the I-Plan update, the 2023 annual report, our TMDL program update from our folks from TCEQ are here to give an update on the TMDL program. I'll have a discussion on fecal indicators coming up.
Opportunity for immediate impact.
And then we're going to have some implementation project presentations from some of our staff here. And then we'll get into some other business and announcements, and then we will be adjourning. So with that, are there any questions or changes or additions to the agenda? Okay. So to determine the quorum, I'm going to just run through the member list. You've got the big roster in your meeting packet for this roll call, and I'll ask Kendall to go ahead and unmute audio at this point. So when I call your name, please let us know that you're here. For those present at the conference room, remember to turn on your microphone and then turn it off. So it's just one tap on that button. If it's off, it's blinking red. If it's on, it's going to blink green. If I call out a big member that is absent, if you are the alternate for the absent member, please identify yourself by stating your name and that you are here for the absent member you represent. Okay, so I'll start. Ralph Calvino.
I'm here. Can you hear me?
Yep. Thank you, Ralph.
Okey-doke.
Chris Backey.
I'm here.
Danielle Ciosi.
Hello.
Tom Douglas. Colleen Gilbert.
I'm here.
Teague Harris. Cassidy Ince. Andrew Isbell. Carol Labresh.
Hi, this is Lisa Montemayor. I'm here as an alternate for Carol Labresh.
Thank you, Lisa. Michael Lee. Kathy McCoy. Paul Nelson. It's here. Well, Scott Nichols. Becky Olive.
Hi, this is Karen Kotke for Becky Olive. I don't think she can make it today.
Thank you.
Thank you.
Mitchell Page. Linda Pahacek.
I'm here. This is Linda.
Hi, Linda. Thank you. Sonia Phillips. Rachel Powers. Jim Robertson.
Steve Hupp for Jim Robertson. He may join us later by Zoom.
Scott Sanger.
Hi, this is Liz Stone for Scott.
Thank you, Liz. Christine Santini.
Here.
Jamie Shakar.
Here.
Thank you, Jamie. Linda Sheed. Linda.
There we go. Here.
Brian Schmafsky.
Brian Schmidt is present.
Hi, Brian. Shane Simpson.
Here.
Robert Snoza.
I'm here. Hope everybody's having a great day.
Thank you, Robert. Michael Thornhill. Scott Tuma.
I'm here.
Thank you, Scott. Joanna Wilson. Natasha Zarnsdorf.
I'm here.
Thank you, Natasha. Okay. All right, well, very good. Based on our members and alternates, we do have a quorum. So move us on to our roster updates. Just really all we have are two alternates to do, and those are alternates for Ralph Calvino, Libby Decker, and Ala Balani. And then we also have Tom Douglas as the alternate for the public. So could I get a motion to approve the addition of these alternates?
So moved. Linda.
Okay,
This
is there a
is I'll
second?
second.
Any discussion? Okay.
I'm sorry, Todd. Can I clarify where exactly is Tom an alternate? I'm not seeing that. Am I missing it?
Linda Bahachik has asked for him to be.
Yes, it was for an alternate for Linda Bahachik for is Tom Douglas. Any other questions or discussion on that? Okay, is there anybody that wishes to dissent to this motion? Okay, hearing none, that's approved. Thank you. So next up is our review and approval of the meeting summary. Please take a moment to review the May 23rd, 2023 spring meeting summary that was included in the packet sent out to you. let's take a moment and do that. If you have any edits and comments, please raise your hand or use the chat feature. Okay, do we have any edits or comments? The minutes? Linda?
Yeah, I know that Tom sent some in, so I don't know, I might be repeating some that he sent in. But I know that one that he probably didn't send in is on item 5, the second paragraph. It says Mr. Tom Douglas. That should either be Dr. Tom Douglas or just Tom Douglas, but not Mr. His appropriate title is Dr. Tom Douglas. And then, Tom, were you gonna say your others that you sent in, or should I go ahead with some of the ones that I think might overlap? I said, are you gonna say the ones that you sent in, or should I go ahead and do some that might overlap with what you sent in? Oh, are you going to say some, the ones that you sent in, or should
I
I?
think they got, I checked a couple that I sent in and they've been included.
Oh, okay. Did you do the timeline ones? Did you do the timeline ones? No, maybe not.
I guess
Okay,
not.
okay, on item 1, the second paragraph, and item 2, the last paragraph, both of those places refer to a timeline that was presented. And I think since not everybody may have been at the meeting, that timeline probably ought to be attached to the minutes.
Okay,
Both of those two, they're two different timelines. I think Tom probably got the other ones that I had too.
okay, with those changes and additions, are there any other comments or edits? So with those changes and additions, could I get a motion to approve the minutes?
So moved, Tom Douglas.
And a second?
Second, this is Colin Gilbert.
Colleen? Okay, so remember, we're voting in the negative. So if you'd like to dissent, please raise your hand to do so in the chat or voice that if you're in the room. Okay, hearing none, that will be approved. Okay, on to public comment. Are there any members of the public or non-members of the BIG who wish to make a comment? Please raise your hand. Now to make a comment or statement. Be heard? Anybody? Okay, thank you. So we're shift gears to the I-Plan update and revision. So for those of you that are new to the BIG, the bacteria reduction plan was officially approved on January 30th of 2013. And we've been in implementation now for 10 years, and we are now actively updating the document to freshen it up with some lessons learned since 2013. We've discussed many of the changes at past meetings. So the focus today is just kind of bringing this update in for landing. So before we get into that, just a reminder about the formatting and changes you will see in the document coming up. There are still 11 strategy areas, and most of those activities remain with some slight changes. The biggest changes are within the activities themselves. We're bringing forward the 9-element table to incorporate metrics, milestones, and monitoring elements within each activity, and the success will be measured on meeting those milestones and improving conditions. And as we bring this document to review, we want to make sure that we're reviewing it with the thought of bacteria reduction, actions that will produce results, and we have a specific topic with that regard on the agenda today. So this document is in process. Really, to finish this update, Stephen would like to form a review committee. That would be no more than 5 to 10 volunteers to assist with the review and act as a liaison with the work groups and the BIG. The invitation will go out to everyone in the BIG, but currently for this group, Ralph Calvino and Brian Schmafsky have volunteered to serve. I want to note that the members of the Joint Task Force will certainly be invited, and Steve is going to get that started in January. And the Coordination and Policy Work Group will also meet in January. And then they will work to prepare a draft of this I-Plan update for the May BIG meeting. Rachel, do you have anything to add to that? No. Okay. So that's where that sits. Some work to do, but want to get with the review committee and go through that and really have a nice polished document to look at in May. Next up is really looking at the update to the 2023 annual report. And I'm going to let Rachel take this portion of the talk and go through some of the information that we collected.
All right, hello. Let's see. You guys hear me okay? I'm gonna move this a little bit closer. All right, my name's Rachel Wyndham. I'm with the Houston-Galveston Area Council, one of the senior planners on the Water Resources team. So I'll just fill in a little bit about kind of the progress Stephen has made on the 2023 annual report. So far, we've acquired discharge monitoring data from our wastewater treatment facilities, Harris County pollution control inspection data, updates to the on-site sewage facility permit data, updates to on-site sewage facility unregistered data. This is kind of our estimation of any unregistered systems in the big area, and sanitary sewer overflow data. So this should be a pretty familiar graph at this point. As we've seen over the past few years, the trend in bacteria reduction has kind of flattened out. But on a positive note, this comes at a time when the region continues to grow population-wise and development-wise. So being at a level point, especially after years of kind of a decrease in bacteria throughout this big area, is kind of a good thing considering all the development and extra kind of anthropogenic pressures in this watershed area. Oh, this was an animated slide. I was unaware. I apologize. Even with these additional kind of areas, we see that the trend is a general flattening in the recent years. All right, up next, we'll talk a little bit about wastewater treatment facility discharge monitoring report data. So we kind of usually go to 3 different categories of assessing this data from all throughout the big region. We look at the number of facilities. So within the big region, as of the 2023 update, we've got 690 permitted wastewater treatment facilities in the project area. Only about 537 of them have bacteria limits. And this difference might be due in part to the fact that you may have a permitted facility that doesn't exist yet. So that could be the disparity in those numbers. In table 2, you can see that there's a general 2.4% exceedance rate for bacteria samples pulled at a daily rate at these wastewater treatment facilities. That still means there's a 97% plus compliance rate throughout this very large area. So again, this is based on discharge monitoring reports submitted by these wastewater treatment facility operators. So this is the data that we have for the region. When we kind of split it up by looking at plant sizes, excuse me while I move this little widget. We do see a little bit of a difference. So still, by and large, really high rates of compliance with the bacteria standard all throughout the big area. I guess when it comes to looking at the daily grab samples, plants at the largest size, greater than 10 millions of gallons per day discharge, they have the highest prevalence of exceeding the daily grab sample rate. But as you can see with a lot of these other sizes, we have a fairly high rate of compliance throughout our project area.
Yeah, excuse me, Rachel, on that previous slide, I know it's an automated issue, but could the plant sizes be listed from the smallest to the largest in order?
Oh, in the report, probably we can sort it out. Yeah, because variable intermittent, we're not sure. We can jump that to the top. Thank you. All right, on this slide, we're looking at table reporting data just from the Harris County wastewater treatment facilities. So the discharge monitoring reports are in the first 3 columns, while the Harris County pollution control inspection data is presented in the other 3. So for the same wastewater treatment facilities, you can see the compliance varies with this Harris County pollution control data. to that of the larger discharge monitoring report data set. And the plants larger than 10 millions of gallons per day discharge here also seem to be at a similar rate of exceeding that standard with the daily samples. So that's probably being captured accurately when we compare it to the full big area discharge monitoring report data. All right, now we'll look at some of the data that we've captured on sanitary sewer overflow events within the big project area. And as you can see, blockages by either fat soils and grease, explicitly that category, blockages by fat soils and grease is the largest single number of sanitary sewer overflow events within this area. Volume wise, we have a pretty large number here in the line blockage for rags and wipes. But I do want to point out that we also saw this number pop up in our analysis of the water quality management plan region-wide data. There was just one report. It was a single day event that reported 75 million gallons of discharge. So that is kind of the downside of using this self-reported data. We're not sure if that's an error, if that was a true observed event, but we just want to take that number with a grain of salt, especially since it's so different from our other categories. It's just that one report might be weighting that entire category. All right. So with that, talking about additional data needs, we're still in need of local data from member organizations and others. So Stephen will probably reach out to those that typically provide those updates and also meet with individual work groups in February to look at additional data. And this programmatic data from member organizations includes implementation efforts carried out. Crashers. All right.
Okay. Hello. Yo. Rachel. Stop muting me.
Okay. This is a first for us. Anyways, a little diversion. We're trying to gather information on implementation efforts carried out over calendar year 2022, any success stories or challenges encountered that could be related to wastewater or stormwater permits undertaken within those programs. So the overall goal, as Stephen said, is to remember to share with one another in the region. So the report timeline for the 2023 annual report is that in January, there'll be the coordination and policy meetings, February work groups. In March, they'll revise the document. In April, the coordination policy review will be called back into order. In kind of late April, early May, we'll send a revised version of this document to the bacteria implementation group, and in May present it to the bacteria implementation group. And I believe that's it on the report. So now moving on to the total maximum daily load updates. I'm not sure from our online participants if we had anybody from the Texas Commission on Environmental Quality that wanted to.
Right here, we got
Okay.
Daniela.
All right.
Hi, can everyone, okay, you can't hear me. Great. I'm Daniela Mejia. I'm the TCQ PM for the BIG, and we've got two updates. One is the Jarbo and Winters Bayou addenda will be going through the approval process and will be approved by the EPA in the spring. And also TMDL will be funding additional monitoring for Tarkington and Loose Bayou in response to its listing in the 303D list. And that's about it. Thank you.
All right, up next, we have our kind of discussion about fecal indicator bacteria. So I don't know, did you want to?
Is- So, is Dr. LaMontagne on the line?
I am present.
Did I miss? Oh, I'm sorry. It's okay. It's hard to see in this cavernous space here.
It's okay.
So we've had some discussion in the past on fecal indicator and looking at some different ways of looking at indicators. And Dr. LaMontagne has doing some work that he would like to discuss with the big, and I think it's something that's really encouraging for us to look forward to. So I'm going to just give the floor to Dr. LaMontagne from University of Houston, Clear Lake.
All right, thank you. The use of fecal culture-dependent methods is kind of perplexing, but at the same time understandable. I'd like to say in grant proposals that we're using a 19th century technique to address a critical water quality measurement. That is culture-dependent methods take 48 hours typically to yield results, and the results don't indicate the risk to the public or the source of the waste. So they're not very actionable. And to address this for the last, I would say, 30 years, there have been different approaches to tracking the source of fecal contamination. Culture-dependent ones, which use libraries and molecular methods. We're doing both. And the reason we do the culture-dependent methods is that is going to be the data that groups like this, water quality managers in general, need to see improvements on or use in decision-making processes. That's not going to go away in the immediate future. What we're trying to do is identify the source by looking at the protein spectra associated with microbes, and we've made progress on that. I think what we're really interested in now, though, is talking about the molecular methods. And those can be broken down into quantitative PCR approaches, which have been widely adopted in the last, I'd say, 10 or 15 years. The most promising method in that regard is cross-phages. These are viruses that affect a strain or a species of bacteria associated with human waste. And they appear to be very good at modeling the viruses associated with human waste that are a public health concern. That is, we want to be able to determine the true risk of exposure to water, and people generally get sick this day and age from viruses in the wastewater, not cholera or other bacteria. So what we want something that models that, and the cross-phages are a good metric of that. An alternative approach is to look at antibiotic resistant genes, and because of the wide use of antibiotics, both in animal husbandry, but also in the clinical setting, the waste from humans has a relatively high level of certain antibiotic resistant genes that have been selected for by the use of antibiotics. And that tracks well with cross-phages. They correlate with each other. And that can provide a second level of indicator. The third approach is to look at human DNA. I don't know a lot about that, but we use a similar approach to differentiate the sources when we find them. Let's say we find scat in the field. We want to know if it's from feral hogs or a dog. We can look at the mitochondrial DNA as a quick method of identifying the source, confirming what we think is the source of it.
What I don't know about the human DNA is if it correlates well with the antibiotic resistant genes and the cross-phages, which, as I mentioned before, agree with each other and support each other. And if there is any issues as far as implementation of the method in terms of collecting and concentrating the filters. And finally, the third generation of methods will be point of sampling methods, where the water quality manager can measure this, identify the source, assess the contamination at the point of sampling. So what we're looking for there are methods that give results in less than an hour without using the resources of a molecular biology lab. So maybe a suitcase lab, a lab that is portable, measure it off on the tailgate of your truck. Those approaches are available, but they have to be married to the appropriate assay. And the way we're leading towards in that is using loop amplification, which is similar to PCR, but it doesn't require a PCR device. And all you need is a hot cup of coffee to keep the sample warm and get it going, and you can read it by a colorimetric change, as I said, at the point of sample. That is a little bit down the road because there are obstacles as far as sensitivity. And again, what actually, if you're going to do that, what are you going to measure? Cross-phages, enterococci, or antibiotic resistant genes, or something else? All of those, I would say, are open questions. What we're doing now is using cross-phages and enterococci with molecular techniques. The approach we're using is analogous to quantitative PCR, but it's digital PCR, which is more accurate, more precise, and shows better correlation to unequivocally contaminated samples. That is, it's very good at detecting fecal contamination, the source of it, with more accuracy and precision than quantitative PCR. In parallel, we're doing a broad spectrum metagenomic analysis to see if there are any members of the microbial communities that could serve as an alternative indicator, particularly in terms of detecting the waste associated or the signal associated with on-site wastewater treatment systems, so-called septic tanks, which might have a quite different signal than the wastewater associated with the wastewater treatment plant. That's an open question, and we need to address that, particularly for work going on here, where in this area, as I'm sure you know, there's an incredible amount of septic tanks for an urbanized area, and we need to make sure that the methods that were developed looking at other systems work for this particular problem. That's pretty much where we are at this time. I can say that our preliminary results show that the cross-phages work, and that there's correspondence between microbial community structure and that indicator of human waste. And I'll yield.
That overview. So we'd like to open up to discussion and questions and just discuss this work.
And Todd, we did have a question from Roberto Vega in the chat. I can read it out, or Roberto, if you'd like to unmute and ask it yourself.
Sure. Regarding the DNA testing, to find source, from the sample, is that, how's that coming along? And I guess my main question is, does that require multiple samples in order to conduct that, source analysis? And or does one sample suffice and you could say, can you test it for all of these things? Because I would assume that let's just say you sample from the channel, you would have multiple types of sources at different degrees in the sample. So if you selected human, you might find human, but it might not. Does that make sense?
Yeah, I would say that addressing those spatial and temporal, the spatial and temporal variation of water samples is beyond the scope of the available techniques, as they still require days of processing to yield actionable data. We don't have what we would need to do that, which is a point of sampling method that's practically real time. That is, if you took a sample in the middle of a channel and we would still have to, we would still have to take it back to the lab, concentrate it, extract the DNA, and then put it in the queue to run with another set of samples that have the appropriate standards. That takes time and costs a lot of money at this stage. We don't have any way of doing it in the field consistently yet. So if we take that measurement, it's very true that we could come back the next week and there'd be a completely different body of water there, or there'd be variation between the spot we sample with depth or longitudinally in the body of water.
So when you concentrate it at the lab, however many days later, and it's being analyzed, does that analysis tell you in this concentrated component there is human fecal DNA and dog fecal DNA and cat fecal DNA? Does it like tell you all of the sources that were available in that sample, or does it, I want to test for humans, is it present? Yes or no?
It's not yes or no, but it's a quantification of human source at this time. We're not trying to determine all the possible sources. It's the target that we're using is specific for humans.
So if human DNA is not present, then it's a no answer.
Human associated DNA, not human genome or mitochondrial DNA, but the DNA of microbes associated with wastewater, human wastewater in particular.
Okay.
So not even the microbes associated with human fecal matter, but rather the microbes associated with the primary influence to a wastewater treatment plant or the effluent from a septic tank.
Okay.
Because let me back and clarify that the microbes in the wastewater treatment plant are quite different from what is in human waste. So that's what I was trying to clarify. And we don't know what.
Well, that just my last question then, or statement would be, so if no human microbial DNA is found, that would be a no for human source.
Right. If the that is, yeah.
Okay.
Within the, within the detection limit.
Right. Okay. Thank you.
Yeah. should say, for looking at alternative sources, we are doing that with the culture dependent methods, because in that case, we're trying to distinguish E. coli or enterococci depending upon the source. And that can give us an indication of what is contaminating the system consistently. And then we could hone in, if that proves to be a problem, on using molecular techniques that target those sources. For example, we have students this semester isolating enterococci from seagull droppings, I guess would be the terms for them, and using MALDI-TOF to differentiate those isolates from other animals and from human waste.
One of our previous big members, Richard Chapin, told me that the city of Houston had done some testing for bacterial DNA in water, and I guess they also did culture tests for the bacteria. And he said they were disappointed at first because they found a lot of dead bacteria. And I said, well, you shouldn't be disappointed at all. That studies have actually showed that the bacterial DNA was a better indicator of human health risk than the live bacteria, which indicated it wasn't the live bacteria that were causing most of the gastrointestinal disease, and that it's probably a lot better because it's, as you mentioned, it's more of a model for viruses, which tend to go through wastewater treatment unscathed much better than bacteria do. So bacterial DNA is probably a better model than actual cultured bacteria. Does that sound right to you?
Yes, I don't know that bacterial DNA, but we look at bacterial viral DNA, which are the phages, as a model, because they're essentially not alive or dead. A virus is just an inert infectious nucleic acid protected in a capsid, and the phage is the same thing. So their survival in the environment is different than a bacterial pathogen, like E. coli can be, or Enterococcus, or I mentioned Vibrios, Salmonella, etc. Those have not proven to be a very good metric for the cultural bacteria, have not proven to be a very good method for the level of contamination that is of concern or currently measured in the developed world. So it's very good for detecting gross contamination events. They would happen with a complete failure of or overflow of a wastewater treatment plant. But when they look at how they correlate to those indicators correlate to actual human diseases, it's not so good. And most of that data comes from things like a triathlon, when they know exactly who went into the water and how long, and they can track them down and see how sick they got, and then look at the water quality, and they say, it's in a strong correlation, but A, they're getting viral infections, and B, it's the water would not be considered extremely polluted based upon the currently used metrics. Hence the need for the new I would say a third generation of molecular methods.
This is Linda Sheed. I was wondering, Roberto, why you were asking about whether or not he was testing for other animal sources, or trying to figure out what other animal sources might be there. I was wondering where that question was coming from, what you were trying to get at.
Well, it sounded like the purpose of the analysis was to determine source of the impairment. And so whether that was a human source or an animal induced source, was why I was asking, are they testing for other things besides humans?
Yeah, I'd like to address that. You're spot on. The source of the impairment is different than the source of a measurement of the risk. And the source of the impairment is how do we bring down the y-axis on that figure? We need to know what is causing those counts, to achieve the stated objectives, which is to be in compliance. That's a different goal than determining what is the true risk to the public, because the risk of human associated waste is much greater than the risk of animal associated waste. And we try to do both. We try to address where the conventional fecal indicator bacteria are coming from with the library-dependent method, which is very good at distinguishing the source of those particular bacteria. How that relates to the public health risk of exposure to that water is not clear because of the poor correlation of those indicators and public health outcomes. So that's why we need a third generation that would be a point of sampling method that directly detects either the pathogens or an indicator that models the pathogens associated with human waste.
Yeah, so the regulatory rules are all based on bacteria, like, as you said, the y-axis on the graph. And so we tend to really be fixed on bacteria, and we count the bacteria. And you commented, if these bacteria were contributed by humans, it would be a lot bigger risk to public health than if they came from, a seagull, say. And they'll count the same on the graph that we usually look at. And so, having been a research guy for a long time, I thought, well, if you want to know if it came from humans, why don't you just look for human DNA? Because human feces, their cells are getting sloughed off your intestine all the time. And there are quantitative assays for human DNA. And I know those don't relate to the administrative standards that we work with. So we have to know, how does this relate to what people are used to? But it seems like if we really wanted to know, we just look for the human DNA. And in response to the other question, there's quite specific assays. If you want to, you can quantify human DNA, you can quantify pig DNA, you can quantify dog DNA, and you can do all those on the same sample and find out if you really think there's a dog kennels a little bit upstream and you think it's a dog contribution, you can find out for sure. But it doesn't, I think a problem that we face is it doesn't relate to what this graph is that we're used to seeing, because it doesn't count bacteria, but it's probably a lot better reflection. I think in the long run, it would be really beneficial to have, well, we have data on counting bacteria and risk to human health. And that could have been done by looking at human DNA and looking at the risk to health. It's just that those studies weren't done, so we have to go with the measures that we have. But I would like for us to keep in mind in the longer run that that's just going, you don't have to rely on bacteria that are associated with humans. Just look for humans and then you'll know for sure.
Yeah. And there are other indicators that one could use that are really beyond my field, like caffeine. You don't find that a lot in dog waste. And there have been much better teams than I could put together that have looked at multiple parameters, indicators of human waste, including chemical, caffeine, as well as stable isotopes, because as our food has a different carbon and nitrogen signature, particularly nitrogen, than naturally occurring, than what a wild animal would consume. And so you can use that to determine if the nutrient pollution, and hence if the contamination to the system, is coming from domestic sources or from wild animals. But that is a real interdisciplinary research program that's beyond the scope of what we could put together. But that type of work has all pointed towards using digital PCR for markers associated with human waste. And the class 5 seems to be the best one right now. And the trick will be to make it mobile, get it into the field, because the impetus, my interest in this really came after Hurricane Harvey, when everyone was asking me as a microbiologist if the water was contaminated, and I was like, they won't let me into the lab. So I was taking samples and running the gauntlet to take them all the way up to College Station to be analyzed. And that was just unacceptable to me. I don't like to drive that much. And, we need methods that are available when we need them most. Not during extreme weather events. And when the risk to the public is high, they don't have to question it. It's not up to them whether they're going to go swimming. They have to wade through that wastewater to get to their house, rescue their cat or whatever it is. They need actionable data at that, within hours. And that's where we have to go to. I'm not sure exactly what the technology will be, but it's certainly available now. That is, you can, when I was in industry for 10 years, I worked on a biothreat detector that could sample the air in this room automatically and identify 20 bioterrorism agents and report the results within an hour over a network. And that was 15 years ago. And I was shocked and dismayed when I went back to look at this field that, we're still, as I say, doing 19th century techniques. Just, I don't get it. But
So,
I will yield.
so getting back to the discussion topic about wanting to have a workshop to try to address these points. It seems to me I don't think this fast, so you'll have to bear with me. So I'm just going to throw out some ideas, but it seems like part of what we have to do is educate ourselves and the public more about the distinction between meeting regulatory standards and reducing human health risk. Right now, the regulatory standard isn't doing it. And so we need to understand that better. And then we also need to understand how they might be paired by some of the things that you were talking about, in terms of being able to see what does the regulatory process really tell you, and what could you do with that and something else to get to human health risk. And then, at some point in the workshop, in addition to understanding those distinctions, understanding those distinctions, understanding the different methods that are available to get to the human health risk, more detailed, more understanding of what you were talking about, and the different generations of analysis testing to the kinds of things and what can be done, what's best to be done, what can be done now, what we ought to be aiming for. And then part of that, what we ought to be aiming for, there should be something toward the end of the workshop about talking about how do we better, I mean, how do we do something about this regulatory environment that's not getting us to human health risk? How do we get a better regulatory environment? How do we, move that ball forward? And I know we aren't gonna, in this, workshop in H-GAC within the context of the state of Texas and EPA and the whole US, we're not gonna solve that problem, but, we need to start having that conversation to figure out how we can solve that problem.
Yeah, I hate to say it, but I think talk is cheap. I think we've discussed this a lot. If you want people to learn about these techniques, they should do it. You learn by doing, and then you really understand it with hands-on experiences. So what we propose is to have a workshop where water quality managers can come in and run their own qPCR reaction on a desktop device that they could, for $20,000, put in their lab and do it themselves if they're so inclined. But you don't, that's my philosophy as an educator, is that once you actually see that it's not some abstract thing that's done by some lab that is out there somewhere, something you can do yourself, and you can understand what is going on much from your hands-on experience, you're more likely to implement or support those metrics. So we tried to put together something last spring at UHCL, but we couldn't. The goal was to have that in combination or as a meeting with the American Society of Microbiology as a workshop co-sponsored by them, but that fell apart. But I definitely want to do that and provide, let's say, 15 to 20 people the opportunity to do this themselves.
So I can't disagree with what you're saying at all, but at the same time, there's two different levels of things that need to go on.
Right.
One of them is that, and I can totally agree with what you're saying, is the hands-on figuring it out. But the other level is the people who are never going to do that, sitting around the table, trying to figure out how we, what's going on here. What do I need to know and understand about these processes? I don't need to know doing them, but I need to understand them enough to know why we need to do them and why we need to have different, a different regulatory environment. So I think we need both. I think we need what you're talking about doing, with practitioners, and then we need the people around the table who deal with the regulatory environment to understand why we need a different regulatory environment. Does that make sense?
It does. I would, but
so I think HBC is more on the policy, regulatory side. You are on the science, figuring out how to make it work in the field with people who have to do it and can understand, can be advocates for it if they understand it better.
Yeah, exactly. I mean, there is an area in between we would call citizen scientists in some cases, where they can collect samples and analyze the data, and the protocols that we're working to would be something that they could implement. We also target, obviously, undergraduates at our university, but we'd like to see if we could develop a program that reaches K-12 students, particularly 11th to 12th graders, to give them the opportunity to learn these techniques. Because if you want broad implementation, you need political support. And one way to reach a lot of people is through their families. You know, they have a kid that has actually done that, and they talk about it with from the bottom up. So I'm for both. I just don't know anything about the political side.
HGAC could probably.
Yeah,
work in that
and
direction.
we're definitely planning workshops for as we enter the third phase or year of the EPA funded work, targeting sometime this spring, which would be the hands-on workshop. And please let me know if you're interested in participating in that workshop. It would be held at University of Houston Clear Lake.
Yeah, I understand that a lot of this work is done by students. And I think that is a really wonderful aspect of the program that you have, is that UH Clear Lake has quite a diverse student body, and we worry about diversity, equity, inclusion. And I think that is really making that connection of having more people out in the community having a feeling, like you said, from having done this, and oh, it's not some distant, vague concept. It's, well, I've done that myself.
Yeah.
I think that's a really wonderful aspect of the work you're doing.
Not a black box that somebody else knows what goes on.
Right. Yeah, I would say that overall, for good or bad, personally, and many people in my field have tried to move beyond treating the microbial community as a black box, where we model the inputs and the concentrations, but we don't really know who's in that box. So we have a concerted effort going on to map by sequencing all the microbes or representative microbes in Galveston Bay and tributaries to field to it, so that we know it's there and we can monitor the changes in that population in the same way that an ichthyologist would count fish species, or ornithologists would count birds. But heretofore, we're saying, let's measure one species out of 50,000 and call it a day. And that one may be dead, maybe alive, doesn't really correlate to anything. So that's definitely the way we're going. I don't know how far we'll get by the spring, but I hope that we can have a method available that is implementable by a citizen scientist and by high school students.
Okay, well, I would say need to move on. This has been very interesting discussion. I would throw out that this might be a topic of discussion for the coordination policy group. And, it'll take more than HGAC to put a workshop like this together, but, involving Dr. LaMontagne, if we could get some TCEQ folks and the folks that are at this table that are on that policy and coordination subcommittee to get together and really plan a workshop for this first part of next year, I think would be a great thing to do. So I would put that out there if we can tackle that coming up in some of our upcoming policy and coordination subcommittee meetings. Thank you very much. Okay, well, we've got 3 project presentations to go over for you all this afternoon. First is going to be on our FY 22-23 Clean Rivers Program and TMDL targeted monitoring project, and Elling Mann with HJC is going to take us through that.
Hello, my name is Elling Mann. I'm a program support specialist for the water resources team here at H-GAC. Today I want to discuss H-GAC's targeted bacteria monitoring project. Before we get into the targeted monitoring project, I'd like to talk about H-GAC's Clean Rivers program, which is the precursor to our project. Starting in 1992, the Clean Rivers program has been in charge of conducting water quality monitoring and assessment to determine the health of water bodies throughout the region. The Clean Rivers program has partnered with local agencies to provide the most accurate and up-to-date water quality information possible. H-GAC does this through a coordinated effort with local partners and the Texas Commission of Environmental Quality. Thanks to this coordinated effort in the use of advanced data management and analysis tools, policymakers use this same data to prioritize areas with water quality issues and implement the management measures that will have the most impact. So how do we determine where to implement management measures that will have the most impact? Well, each year the Clean Rivers program routine monitoring data is analyzed as part of the H-GAC basin summary / basin highlights reporting process. A main theme to these reports is that bacteria continues to be the most prevalent pollutant within the H-GAC CRP basins. When analyzing this data, our routine monitoring, we examine various watersheds with elevated bacteria levels to identify potential sources. It's at that point we begin taking steps to help manage bacteria levels at these locations. The Clean Rivers program is one of our sources of funding for our targeted bacteria monitoring. Our goal was to identify assessment units that had high bacteria levels. H-GAC ranked waterways using the highest geomean relative to the state standards for contact recreation. The criterion for E. coli is 126 MPN over 100 milliliters, and the single sample criterion is 399 MPM over 100 milliliters. Assessment units were selected by the highest geomean identified and accessibility and feasibility of the waterway for field investigations. This slide shows you where in our region each of the 12 assessment units are located. They're mainly located in the north, northwest, and western portions of Houston. With the 12 assessment units identified, we began our desktop review. Our desktop review helps us gather further details about each assessment unit. We started off our desktop review by utilizing ESRI's ArcGIS applications as well as Google Earth imagery to identify locations of wastewater treatment facilities, permitted discharges, outfalls, and potential sources of point source and non-point source pollution that may contribute to bacteria loading in the stream segments. Other potential sources of bacteria identified during the desktop review include permitted and unpermitted on-site sewage facilities, sanitary sewer overflows, hobby farms, homeless encampments, commercial and industrial facilities, bridge crossings, and other entry points were identified to provide access into the stream to collect bacteriological samples. Once our desktop review was complete, we then began windshield surveys. H-GAC and its subcontractors, the Environmental Institute of Houston, EIH, at University of Houston Clear Lake, and the Texas Research Institute of Environmental Studies at Sam Houston State University was conducted a windshield survey on each watershed. Field crew collected bacteria samples at easily accessible locations, such as major road crossings and public access points adjacent to the waterway. This survey served as a spatial assessment of the watershed and determined where hotspots of high bacteria concentrations existed along the waterway and its tributaries. Results from the survey aided in prioritizing intensive field investigations along the waterway and tributaries of concern leading into the main segment. The windshield survey was only conducted during dry weather following a 72-hour period. After the windshield surveys were complete, field investigations took place. The field investigation was a survey where a team of 2 walked the assessment unit and sampled any waterway observed flowing into the stream. Water could be flowing in from a pipe, culvert, natural tributary, or earthen ditch. Flowing water was categorized into 2 source types: permitted outfalls and unpermitted outfalls. Permitted outfalls included wastewater facilities and municipal separate storm sewer systems. Any pipe greater than 12 inches in diameter was assumed to be permitted by our field crews. When flowing water was observed from permitted outfalls, 2 samples were collected. 1 sample was collected immediately downstream of the outfall, where the flowing outfall was mixing in with ambient water. The second sample was taken upstream of the flowing outfall outside of the realm of influence from the outfall to provide the ambient bacteria levels of the assessment unit in the area. Unpermitted outfalls, they were categorized as any other flowing source of water that was judged to be permitted in the field, including small flowing pipes that were less than 12 inches in diameter, and tributaries. Tributaries were sampled directly with a single ambient sample taken upstream of the confluence of the main channel. For investigated reaches where no outfalls or tributaries were identified yet showed elevated levels of bacteria during the windshield survey, ambient samples were taken by bracketing locations to narrow down where unseen bacteria sources might be present. The field investigation was only conducted during dry weather following its 72-hour dry period. There were 2 field investigations that took place. H-GAC conducted its first field investigation on April 20th, 2023, 12 days after significant rainfall within the watershed. For the first field investigation, 15 samples were collected at 11 locations. Outfalls were an upstream and a downstream sample were collected, are noted in the sample ID with a U or D respectively. 5 locations were identified and recommended for further investigation by the field crew. The second field investigation was conducted June 29th, 2023, 8 days after significant rainfall. The second field investigation was conducted to identify and reinvestigate sources from the first field investigation that had high bacteria results. 11 samples were collected at 11 locations. There were no observed flowing outfalls during this field investigation, and the tributary investigated was experiencing intermittent flow conditions. Of the 11 samples, there was 9 that reflected a bacteria level greater than the primary contact recreation single sample criterion. While nearly all the samples collected exceeded the single sample criterion, 3 locations were recommended and investigated based on either their high bacteria levels on the tributaries or substantial changes in upstream and downstream ambient sample bacteria results that suggests a source that was not located during the initial field investigation. When we were in the field collecting data, we utilized ArcGIS's Survey 123, a cloud-based application that allows the users to create a custom form and surveys. In addition, it allowed water resources staff to see exact locations of samples collected by the field crew. Field crews are able to collect data using a tablet even when disconnected from the internet. Data is uploaded securely and results can be analyzed on ArcGIS app. By the end of the project, we gathered quite a few samples. We had 108 samples in all the windshield surveys, 587 samples collected within the field investigations. 109 sites were included among the referrals to the proper authorities. In addition to referrals for specific outfalls, some referrals included areas where high bacteria levels were found in ambient samples without any flows observed from nearby potential sources. Each assessment unit has an individual report detailing methods and findings, which has been made available on our website. After the project is completed, our ultimate goal is to take that information and use it to help mitigate bacteria levels and improve the quality of our region's water. One of the first steps we take is sending the referral sites to the proper authorities to see if any potential sources can be eliminated with repairs or corrective action. We also see if any of the referral sites resulting from the ambient reference samples can have their sources identified. H-GAC anticipates future iterations of this project with a reanalysis of the priority assessment until it is listed based on more recently obtained ambient monitoring data through the Clean Rivers program. With future funding, we also anticipate that some of these assessment units will undergo additional field investigations, and in turn, we will continue to work with the proper authorities towards corrective actions. That's all I got for you today. I have my contact information listed below if you have any questions. Thank you.
Any questions for Elling?
I saw that, Tom Douglas, I saw that we had Colleen Gilbert on today with the Greens Bayou Coalition. Colleen, if somebody wanted to access this data or Ailing, is there a way that Colleen or somebody with a specific interest in Greens Bayou could address this data?
Yes, we have one of our assessment units that we did our targeted bacteria monitoring on. It is one of the assessment units that we did it, and it is all available on our website, and I can send you an email to that link.
Great, thanks.
Thank you very much. I would very much appreciate it. We're doing some sampling now, and we've got 2 people right now on our team, a very small team, but we're working on it.
Okay, up.
Yeah. Hi there, Taylor Chapman. As of recently, the Bayou Director for Friends of Woodland Park. So Little White Oak Bayou runs through Woodland Park. I just got trained up by Grant at the Stream Team program, so I'm excited to plug in. But I would love to follow up in particular on the 2 impaired sites that are tributaries of Little White Oak Bayou to see if there's anything we can do locally. We've got lots of folks from the neighborhood involved, and so happy to help chase it. Just would love to get involved.
Awesome. I can send you some information regarding that.
Super. Okay, up next, we've got Kendall Goodrose, and she's going to talk about some of the same type of targeted monitoring, but what we've the monitoring been doing through one of our grants for the Galveston Bay Estuary Program, working with the Biopreservation Association. Turn it over to Kendall.
Sorry, I shared my screen and then my mute button disappeared. So, all right. So, good afternoon, everyone. I'm Kendall Gaudreaus with, again, with the Water Resources team here at H-GAC. And I'm going to provide just a short update on our targeted bacteria monitoring project. Several of you have heard about this project before. It's had several phases over the past few years, but this project is a little different than the one Elling just talked about, though the goals are the same. And this one was done in partnership with the Biopreservation Association with funding from the Galveston Bay Estuary Program. So just starting off, if you haven't heard about our project in the previous phases, what this was an attempt to not only do targeted bacteria monitoring, but also to do it in a way to test out whether a design could be more cost-effective to help identify those potential sources that might be in need of corrective action to reduce bacteria in our waterways than the professional monitoring where the analysis is done by laboratories, the way the project Elling just discussed was. So for this project, H-GAC provided technical guidance and project oversight, and like I said, partnered with Biopreservation Association, who did the actual monitoring with the help of interns. So for the first 2 phases, interns were recruited from the Student Conservation Association. And in those phases, each set of interns, you had 2 interns for an 8-week internship period, which kind of threw up some roadblocks, which I'll talk about later. And then for this last and most recent third phase, which the update's going to focus on, biopreservation actually tried to overcome some of those roadblocks by recruiting interns directly for a longer period of time. But it seemed like this past year it was a little hard to recruit interns just kind of across the board. So they ended up instead partnering with a Texas Conservation Corps team who helped staff conduct that monitoring. So we were still able to get those interns involved, and we had some more hands out there trying to help us complete the monitoring. Just like the other project, this one does follow an approved quality assurance project plan. But this project actually uses Texas Stream Team E. coli monitoring protocols to determine those bacteria levels, which is great because the interns who can get experience not only get to do the sampling, they get to do the analysis of the bacteria as well. So it's a cheaper for materials to do that analysis, and you get to do it kind of in-house, as it were, so you can get those results a little bit faster. So in this specific project, we also focused on waterways from the big. We had 6 AUs or assessment units that we focused that were either in the top 10 list or directly adjacent to the top 10 list. And like with the other project, we were trying to find things like illicit discharges, sanitary sewer overflows, broken infrastructure, or other potential bacteria sources. This is just a map of the 6 prioritized waterways. And as of this August, all 6 AUs had been monitored throughout the project. As with the other one, that was in dry weather sampling where it hadn't rained or hadn't had any significant rainfall within 3 days. 4.5 of these AUs were monitored during the first 2 internship rounds. Between December 2020 and July 2021, and the rest were completed over this spring and summer. So as I said, the AUs were selected from the BIG's top 10 list, or just outside of it, and they were also kind of determined and prioritized based on their proximity to the Biopreservation Association's old office, as well as the length and feasibility for interns to actually complete that monitoring during those periods of time where we had them. The ones that I'm going to focus on today that we just completed are actually Pine Gully, which is in the bright pink color, and the remainder of Little White Oak Bayou, which is that watershed in the brown color. In total, 113 samples were taken from the 6 AUs across the entire project. With 71, or about 63% of them being over the state's single grab standard of 399 colony forming units per 100 milliliters. 13 of those, or about 10%, were over 20,000 colony forming units per 100 milliliters, which was the upper limit of the testing method used for the Texas Stream Team protocols. While that is really just a high level look at the sampling efforts and what we saw, I did want to dig a little deeper into a few of those AUs where we received feedback from the city of Houston. Big shout out and thank you to Lisa Montemayor for working with us on this project to help investigate our findings. So of the AUs you see on your screen, unfortunately for Country Club Bayou and Berry Bayou, those were sampled in the summer of 2021, which you may remember was quite a bit wetter than average. That made it very difficult to not only get dry days where they could go out and do the sampling, but it also kind of prevented a lot of follow-up investigation, since once it rains, it kind of changes what they're able to investigate for the findings that the interns had. The unnamed tributary of Buffalo Bayou is also kind of a special case in that the only parts of the AU that are accessible are in Glenwood Cemetery, but based off of our findings, it seems likely that lines and sources could be coming from nearby neighborhoods. But again, due to that limited above ground access, wasn't necessarily possible to identify anything more directly. However, as I'll show you in a second, the interns did find and report an incidence of residue at that AU, which turned out to be fats, oils, and grease, suggesting again, sources are likely from the nearby lines coming from neighborhoods. For the other 3 AUs, Plum Creek, Pine Gully, and Little White Oak Bayou, City of Houston Public Works was able to investigate the results and provide us with their findings, and I'm going to go over those with you today. So first, that is the fats, oils, and grease residue that was found at the assessment unit in the cemetery. Fortunately, that's all they were able to find there, but it is kind of interesting to see. But in terms of Plum Creek, as you can see, this had some of the higher results, with 3 of the samples being over 20,000, so over that testing limit. And what was found was that an SSO that was actually found and reported by the interns was likely responsible for 4 of the high results. They also found a potential sewer leak at an apartment complex, which was likely a possible source of one of the really high samples that was over the testing limit. It was referred to the health department. A possible private line issue was referred to flood control. And then again, an additional sanitary sewer overflow was likely the cause of another of the very high results. And another likely came from a small storm line that unfortunately, when they went back, was dry, but was likely believed to be an issue. It may be one of the nearby businesses that drained into that outfall. This is a picture of the sanitary sewer overflow that the interns actually found and reported near Plum Creek. And this was something they just found. They were doing the site survey. They saw what they thought was a small tributary, and with a very bad smell. They followed it up and were able to report that it turned out to be caused by a damaged gravity main line. For Pine Gully, that one was actually completed this spring into early summer. The 2 sampling events this summer did not have quite as high results. We didn't have any of those over the 20,000 colony forming unit levels, but we were able to identify several potential sources with the help of Houston Public Works. So a sewer overflow, again, at an apartment complex was identified, and blockages were cleared from that. Another sewer blockage and potential structural issue was found. They found a blockage on a sewer gravity main at a manhole. It was a grease blockage. There was an underground water leak identified, possible private sewer issue that was referred for further investigation, a water leak, and then another referral to the health department. And then for Little White Oak Bayou, this was not the entirety. This was actually partially done in the 2021 internship period, that wet summer, and it was finished up this year. And this year, this summer, just in those 25 samples, they were able to find that results were likely due to an SSO that had occurred 3 days before sampling, a collapsed sewer line, a possible private storm line, and a sewer overflow identified at that apartment complex. I think you might be picking up a couple of themes in these findings. Oop, too far. So throughout the 3 phases of this project, we did hit, as I mentioned, several roadblocks that prevented an earlier completion of monitoring and kind of the post-investigation sampling that we had originally wanted to do. I mentioned that unusually wet summer in 2021 that corresponded with the second internship round. And then On the opposite side this year, you're all likely familiar with the unusually hot and dry summer we had. The dry days meant that finding days with the required 3 days without rain wasn't difficult, but it did reduce the amount of time that the samplers could safely spend each day in the field. Pine Gully, in particular, went very slowly this summer, as monitors also had to contend with the ragweed jungle that you see on your screen, as there was excessive vegetative growth all along the banks. And then I mentioned they had some difficulty securing interns this year. But one of the biggest roadblocks we had in this project was actually supply issues. You can see on the screen the Coliscan Easy Gel Petri dishes that are used to do the bacteria analysis. And since the beginning of the project, there have been some issues with the manufacturer just having in several instances, there were gel where the bacteria colonies did not change color, which is how we identify what is E. coli. Some didn't gel at all. And this is actually an issue that is going on statewide, so that this Texas Stream team has continued to have those issues with Coliscan for a few years now from the manufacturer and are investigating potential alternatives for Texas Stream team protocols in the future. So while that has been an issue for this project, it doesn't mean that this process of using Texas Stream Team protocols and interns or other organizational methods isn't still a good option for a low-cost framework for this program. And then even with these roadblocks, a lot of valuable information was still collected, including some of those lessons learned that can be passed along to other entities interested in similar programs, because through this project, we wanted to kind of test a model that can be implemented by any organization or entity, with adaptations as needed to fit whatever structure they were looking at. One of the biggest areas we've seen that might benefit from different structures is the intern schedule. Some of the issues we had with weather and supplies and even recruitment might be alleviated by implementing a continuous intern schedule that would not restrict monitoring efforts to a short time frame and would help provide more immediate follow-up if investigation takes a little while. Plus, it would allow for more targeted outreach as part of a larger program. In particular, if the internship was paired with other duties, it might draw in more candidates and make schedules more flexible. In terms of our results, like I said, I think you may have noticed some themes. One key lesson learned, as you saw, is that several sources are suspected to be related to sanitary sewer overflows and blockages. Kind of throwing back to what Rachel showed about the data we have, a lot of sanitary sewer overflows and fats, oils, and grease related blockages causing some of these issues. Along with those other potential sources we found, it kind of just highlights that some of these things really need that kind of targeted education in these communities, so that while monitors can't be out there sampling every single day, if residents know what to look for and how they can reduce these instances, it can still help reduce the bacteria that is entering our waterways. So with that, I will see if anyone has questions.
Any questions for Kendall?
Yeah, so some of the sources that you cited were sanitary sewer overflows. And one thing I always wonder about is, how good is the reporting of SSOs? Were the SSOs that you found were associated with your high levels, were those all reported, or did you discover some of those?
The interns did discover that one during that first round. So they did discover and report the one that was responsible for 4 of the high values. but I believe all of the other sanitary sewer overflows, all of that information came from Lisa, so they had been reported or found by the city.
Hi, Kendall, can you hear me?
Yes.
Hi, it's Lisa. Hey, I can answer that question.
Thanks.
Yes, all of them, including the one that the interns identified, from the structural issue near Plum Creek. All of those were reported to TCEQ. And we've really, in the last couple of years, have tried to diligently identify, and report every single SSO, though, even those caused by private issues, even though those aren't necessarily reported or required to be reported under our permit, we're still tracking all of those because we absolutely want to make sure we have that data available for analysis, and to be able to determine where our problem areas are, our hotspots, et cetera.
Thanks, Lisa.
No problem.
Taylor here for Little White Oak Bayou. What's the best way to follow up maybe after to get more information on the findings on Little White Oak and how we can follow up? Is it Kendall or who? Either Kendall or I think just sent out a few messages. Excuse me. Either Kendall or I work. Jessica Cias is our data manager. She also has information on that, and she is actively sending out data logs as long as summary reports for all the AUs. So she can, if you reach out to any of us, we can get that to you. Thank you.
Okay. Thanks, Kendall.
Mhm.
And then next up is Rachel is going to talk about watershed protection planning and implementation that's going on HJC.
All right. Hello again. I don't have any slides for you guys. I was just going to give you a quick update on a few of the projects that we're fortunate enough to have funded through the Clean Water Act 319 program. So we've got one project that's basically at its end, the Clear Creek Watershed Protection Plan Development Project has completed their draft watershed protection plan, and it's gone through its public comment period. So that's now off to the Texas Commission on Environmental Quality for further review. Hopefully then it will move on to the Environmental Protection Agency for final acceptance. And we should be able to update that stakeholder group, hopefully, a little bit into 2024. We also have a project going on the East Fork of the San Jacinto River. And that watershed area is a little bit north in our region. It covers a little bit of Harris County, close to Lake Houston, as well as parts of Montgomery, Liberty, San Jacinto County, and also Walker County. That project has also completed a draft watershed protection plan, but the public comment period is open currently. So if you would like to review that plan and submit any public comment, that period is open through December 22nd, which is a Friday. If you can just get those comments to me, and all my information is provided on our project website. That's also where you can find the actual draft document at eastforkpartnership.com. Just provide those comments by end of day on Friday, December 22nd, and we'll incorporate those into our watershed protection plan. We also have an implementation project happening in the West Lake Houston Basin. So this covers the watershed areas of West Fork, San Jacinto River, Lake Creek, Spring Creek, and Cypress Creek. So for all of those watersheds, we've already developed watershed protection plans with those stakeholder groups. And now we're moving into the implementation phase. So we did just complete a data analysis report. So we actually have something that we can share with the stakeholders, a little bit of an update since our last activity in the watershed, which was about 2021. So we can kind of fill you in on what the current water quality status is right now, and some of our plans for some of these implementation dollars, mainly in that area. We saw pretty big signal for pet waste contributing to those water quality impairments. So one of the strategies that we'd like to move forward with is working with our local partners to install some pet waste stations out in those watershed areas. We're hoping to host some public meetings as early as January of next year to kind of get into the details of that and talk about some other implementation that we might be able to undertake in those watersheds. And then finally, we're any day now starting work on the Greens Bayou Watershed Protection Plan development for Greens Bayou and also Halls Bayou, the tributary to Greens Bayou in kind of the northeast Harris County area. We're kind of waiting on a few last administrative things with the contract to be approved. And as soon as we're off the ground, we'll start work on analyzing the water quality in that watershed. So we have something to bring to our stakeholder group. Hopefully, we call some stakeholder meetings on that project towards the end of summer of 2024. So we're excited to work in that watershed. We know we have great partners with the Greens Bayou Coalition, so we're excited to get moving on that one. And that pretty much concludes my updates for Watershed Protection Plan projects. Does anyone have any questions for me? All right.
Hey, thank you, Rachel. So that brings us to, I guess, upcoming meetings and events. So lots of things going on here. December 12th, our CRP Regional Monitoring Work Group has a meeting. Of course, that's on Teams. We take time on this December meeting to do a little bit of training with our local partner agencies. So that is going on. And then on February 9th, our Parks and Natural Areas Awards ceremony will be held. And you can join us for that Natural Parks and Natural Areas Summit as we celebrate 23 award-winning projects that serve as models for planning and project implementation for parks and natural areas in the region. Projects will be honored in 4 categories: planning process, on-the-ground projects over $500,000, on-the-ground projects under $500,000, and programming. Participants will also hear about equity in parks and natural areas planning from keynote speaker David Buggs, the Chief Diversity and Inclusion Officer for Texas Parks and Wildlife Department. And you can register at www.h-gac.com, Parks and Natural Areas Meeting Materials. Somebody may want to put that in the chat. Also working to schedule a Clean Waters Initiative workshop on indicator research in September, and then a Clean Waters Initiative workshop on wastewater treatment facilities and collection systems. And then we've got some public meetings coming up. February 26th, the Big Creek TMDL. February 27th, the Oyster Creek TMDL, a public meeting in Lake Jackson. And then, of course, a big one, March 23rd, is the 30th anniversary of the River Lakes Bays and Bayous Trash Bash. So we hope you'll come out and join us for that. And of course, for this group, the next meeting is May 23rd for the big spring meeting. Anybody else on the group have any announcements they'd like to make?
Excuse me, Ted, could you clarify the agenda says the next meeting is May 21st?
Let's see. Is that a Tuesday? Hold on. It's the 21st because it'll be a Tuesday. May 21st. Thanks for catching that, Danielle. Any other announcements or things coming up from other groups? Like to announce?
Hi, Todd. This is Christian Rhines with the Galveston Bay Estuary Program. We have a couple of our subcommittee meetings coming up this month. So our natural resources uses subcommittee meeting will be tomorrow from 10 to 12. The monitoring and research will be next Wednesday, the 13th, from 9:30 to 11:30. And then that afternoon, the water and sediment quality subcommittee meeting will be from 1:30 to 3:30. These are kind of important meetings. We're going to be discussing updates to our Galveston Bay plan. So anyone that's interested in helping us out with that, we'd appreciate if you attend. If you need any meeting links or anything, just shoot me an email. Thank you.
Great. Thank you, Christian. Okay, anybody else?
Yeah, Todd, I just noticed the photograph you're showing there. I think that was a really good example of, we were talking about the importance of hands-on experience where people in the community can actually understand how these things are done. And I think that was a really fine example. Wasn't that taken at Jersey Village?
Yeah, that was the Jersey Village Clean Waters Initiative. Yeah. And we got out that day and just kind of went over Clean Rivers program monitoring, how it's done, the equipment that is used, and try to get people to come out and just kind of makes a difference for people to be able to see what how things are done. And actually, while we were out doing that, we had a couple residents from the community came over to see what we were doing, and they're able to express some of their concerns and things that they've seen. So it's always helpful when we get out and do hands-on things like that. Okay, well, with that, if nobody has anything else, just again, we thank our TCEQ TMDL program for their support and continue to fund us and do this great work and work with all of our partners. So with that, I guess we'll be adjourned. Thank you all.